That's odd. It's not showing me a picture here. It's not. That's crazy. Okay. I'm having some technical difficulties with the movie I made. Whatever. I don't know what's going on. Anyway, can you see that on the other channel? You guys go there. I probably won't get anybody seeing this till tomorrow, and it'll make world news. My camera's on here. All right. So, I need to get my lighting right. I'll let everybody filter in here, and well, I gotta go to the bathroom real quick and change my lighting here. Get lights on Monkey. I'll be right back. Alright, shit. Alright, shit. Somebody's got my footstool. We are live, fuckers. Digital. Yeah, I see you, digital. I can't get this any straighter. I don't have a fucking holder for this thing. There, is that better? Go Go to Not Your Father's Country channel, digital. That's where I'm at on the computer. That's where the show is going to be. As soon as everybody starts filtering in here. Okay, we can get rid of that. I got 3 people here. Turn my lights down a little bit. All right, so I'm a little tired from doing all this research, guys. I mean, I've got a whole tabs full for this one, for the COVID one, and then I got a whole another browser tab to bring up for the CERN one. So it's 6:19, and we'll probably be in here till well around 9 o'clock. 6:19 my time. You know, so I mean, and I got some notes. I printed out stuff. So yeah, there's a little sneak preview of. I got a whole bunch of printed out of the frequencies. So it has to do with CERN and the frequencies, that the new frequency that's popped up on the VLF, and and if you want, you can correlate it all on the CERN, the web tools, CERN's web tools, Vistars and shit. Soup, White Boy, welcome. Who do we got? Who else we got in here? We got one on Twitch. My main follower on Twitch there. Loyal follower there. I don't know who it is. Could be management, I guess. Because I'm an awful fucking slippery guy. A wiener is, anyway. Pam, hey, what's up? Okay. So what I was trying to do, first, what we're going to do is let everybody filter in from my Mr. Mission Control channel and pull some people in. And you guys are I'm going to pull chat up here for you. Because I had music set up for like, my regular intro music to the live stream, but tonight's going to be a little bit different because tonight's a special. I'm running Mr. Mission Control channel on my phone live streaming, because I just found out I could live stream from my phone. So that allows me to live stream on YouTube from my phone and hopefully get super chats. Right? Instead of having to go through my PayPal and donate. You can super chat on my Mr. Mission Control channel. Just hop on over there, throw me $5, come back over here, and get the information that's worth $500,000 million. What I'm about to reveal to you guys, because you'll be able to, if you had the money for the lawyer, or if you know your lawyering skills, you'll be able to take whoever's responsible for all this to court with the information I'm about to bring you. And it's all public access. There's no dark web shit, no hacking involved. It doesn't take a goddamn rocket scientist to figure out what I figured out. And point it all together, and it's right there in front of your eyes for everybody. Tonight is going to change. You're going to change your pants tonight, maybe 3 or 4 times. You're going to change your pants. All right? Because you're going to piss them. You're going to shit them. You might throw up a little bit. I mean, this is some heavy slap in the face, because nobody can avoid this. Nobody can avoid this. No, we haven't been able to avoid it for CERN for since they've been online, and and the COVID-19 since its onset in the beginning of 2020, or really December of 2019 here in the United States. I know my area was one of the first places to get it here in Walmart headquarters territory. Everybody was sick. I mean, it was worse than the lockdowns. The end of December or January, all of January and most of February, it was worse than the summertime and the lockdowns when you weren't supposed to go anywhere. There wasn't nobody going anywhere, dude. We were all sick. We were all fucking sick. And if people tell you there's no such thing as COVID, they're lying, dude. It's a bioweapon, dude. It's a bioweapon, and I'll show you how it's a bioweapon, and that it's not only a bioweapon, but it's a CRISPR. You all know what CRISPR is. It's a CRISPR bioweapon. It's not what you think it is. It's not what anybody thinks it is. I'm going to change that viewpoint tonight. I mean, to the viewpoint that COVID-19 has got CRISPR in it. They're experimenting with the variants with CRISPR technology. The macrophage, what is it called? Mycobacteriophage. Okay? We're going to get into all this shit. We're going to get all this fucking, it's going to be wild, dude. We were running 13 minutes here, and we're running, I don't care the time limits, because really, if these other platforms can't fucking upload my shit right, like BitChute, well, they're not going to get this content, because this content is worth people's lives. I mean, worth millions of lives. It's worth your life. It's worth my life, and that's priceless. This information is, I don't know how or why it's even on the internet. I don't know, because they have to have propriety for their stuff to make money off of, right? If it wasn't for money, you wouldn't see this. They would hide this stuff if it wasn't for money. If it was something else that we survived on, like Loosh. If we survived on the Loosh, of course, I'm sure back at home world or wherever the fuck is going on, or whatever dimension we're from, they're not showing the population what's going on here. The kind of fuckery that's been going on for well on probably 300 fucking years. This is the real serious shit. I mean, the real, like, you stab your brother and uncle in the back and burn their children and shit, for a fucking postage stamp with a train on it that you're not going to mail. You're going to keep. You're going to save it. I just right off the top of my head, but I was kind of waiting for my buddy to come by so I could show him. When he comes by, I'm going to pull him into the stream and show him this. But I'm going to have a real quick smoke because I've been prepared. I mean, I've been nonstop just preparing everything. It took me a whole day to get this computer line back out after I had taken my break, Paula Shields, hello. And I got Pam, I got White Boy, I got Soup in here. We've got 5 here on Not Your Father's Country channel. And I'm streaming, of course, to all those other platforms. I got 1 on Twitch. 4 on my not Mr. Mission Control channel. But you guys are going to want to come over to Not Your Father's Country channel. Just, it's on my main page, profile page there, where my channels are up the top. Or the Technopathic Technomage. I was going to stream a Technopathic Technomage, but nobody's got that subscription yet. And you can't find that channel because I got the fonts so fucked up, crazy, that you have to copy paste the actual how I got it to find it. Because if you search technopathic technomage, you don't find it. Because I tried. You have to have the link or something. So that's really a hidden backup channel. But I want to get my intro music out of the way because I did find some good intro music. Really, this goes for... Really, that goes for the intro to the... okay, no. I got an intro for the... I just don't have any outros. Well, yeah, I do. I got the 24-hour live stream outro. So we could have a dance party. We could have a techno party, a live industrial dark techno party, Revenge Radio afterwards if you guys want to. I mean, we'll just get some weed, get some speed, some fucking beer, and fucking and camp out all night. You know, with Monkey back here, he's got his pipe and shit. He's already half cocked over, high as fuck. You know what I mean? So he's staying here all night long. I don't know about you guys, but he's staying here all night long. I'm wound up like a fucking motherfucker. I'm about to take me a testosterone pill at Vyvanse because Psychonaut Portal is here. Almond's here. All right. So I got everybody. You guys got to chill to some music for a minute while I go take me a vitamin and get a cigarette real quick. Well, that's only 3 minutes, though. So you guys are going to have to listen to the 24/7 techno. And then I'll run the little intro. You're going to like it. You're going to like it. But just, you'll have to chill to this and chat in the chat room. Get everybody over here from mrmissionctrl channel.
So let's say you have to read an incredibly long email from your boss that you have to finish before the big meeting.
This technical difficulty is all the time. Oh shit. knows that the early bird gets the worm, but the early bird gets the worm, but everybody knows if you're on my channel, if you wait until I'm ready to fucking get shit in gear, you get the good shit. It's like any fucking old fucking drug dealer. Because if you were waiting for old Joe, old GI or whatever, waiting for the van man, they called me. If you're waiting for the van man, he is coming. He's coming. He's coming to the party with, and he's got whatever you want, man. Usually, shit you never had before. Shit you never had before, man. Is what I was packing. So, all right, Friga's here. Friga already knows everything. She hasn't seen it yet. Well, she hasn't seen what I had on the computer, but some screenshots. And then I got to get her some pictures over to her. Now, Psychonaut's still in here. Okay, we got 4 here. We got 2 on Mr. Mission Control channel. You guys on Mr. Mission Control channel need to come over to Not Your Father's Country channel. Wait, how is your chat showing up in Mr. Mission Control channel? Sean and On, what is this shit? This is I'll show you. No, I won't show you yet. I ain't ready yet. Chill out. I'm going to cut chat, and we're going to get into it here. So where should I start? So anyway, for my last live stream, we knew we were looking at the RxN mapper from IBM and the IBM COVID-19 knowledge graph, the IBM RxN for chemistry, where you make your own chemicals, your virtual chemicals. So I was looking around the SARS-CoV-2 coronavirus NLM on the NCBI, the NIH.gov. Okay, this is NIH.gov. YouTube, get people. I know you're watching. This is from NIH.gov, which is directly linked to this, where the CDC gets their shit. I mean, it all comes through here first, guys. It all comes through here first. I've even got the gematria on this shit. All right? You're going to trip when you see the gematria. Well, we're going to save that for last. You guys waited 33 minutes. So let's get some stuff. Leslie just got home. See, people are going to be filtering in here all night, because I usually don't go live until 7 o'clock, and it's 6:45 now. So this is just kind of like fucking just hold your horses. Let's everybody get a drink, because you're not going to want to put your phone down or get up from your computer or your TV after I get started here. That's how fucked up weird it's going to get. Frigga, tell them how. Frigga, tell them how just reality shattering this is. I mean, no matter how it's presented. The NIH link in the chat. I'm gonna drop all the links in the Discord. Let me get everybody get the Discord link real quick. It's in the description of this video, but let me get it real quick. I'll post that in chat, because all my links go to my Mr. In the text channel Mission Actual on the Mission Control server on a Discord. So what I do is I should be able to post it on. Shit, why didn't it? What's that? Oh, that's my Aces. I should be able to post it here. And it come up integrations, channels follow, channel following fact, spotting announcement channel. That's not it. How? Fucking A. View channel. Main show channel. That should pop up in chat. Did it? It didn't. It didn't, did it? I don't know how I linked the damn. There's the Discord where the links are going to be, but you all remember how I was posting in Discord and it'd pop up here, and then whatever pops up in this chat would go to my Discord? I don't know how to fucking connect that back because they took my old Discord away. And I don't know. I'm not going to fuck with it right now. We're going to start. There's the Discord, and I'm going to post all the links there right now before we get started. To both parts. There's going to be a lot of links. We need some background music. We need some background music. There's all the links to this part of it. You guys want to follow along or skip ahead and go home early? I don't care, but you'll miss my fucking coverage of it. Let me get you the other part of the show's links. Well, let me save the other. That wouldn't be fair. Somebody would jump ahead and make a fucking stream ahead of me with the other show's links, because that's a whole nother show. The CERN part. The CERN part's a whole nother show. But those links are up on Discord. Twice I got them up on the Discord. Party Central. Who's in Party Central? Oh, shit. Oh, shit. Well, it says my Restream bot's integrated, but I don't see my Restream bot. I don't understand. I don't understand. We should be conveying all the messages over here. You never had a live audience to draw energy from. Okay. Leslie with the tones. Leslie's going to tell everybody about the tones. I don't have any tones going right now, but I should. That's good enough. Loud enough, I guess. Can you guys still hear me? I have to have some music here. We're going to get into it here. And I am going to cut chat so the screen's big enough you guys can see everything. The bass is loud. Bass is too loud. Is that better? Oh shit, what happened? I gotta put my computer on fucking turbo, I guess. I figure I pick up that ultrasound frequency coming from my computer because it's on turbo. You guys getting this? Okay, you got, yeah. We don't have volume up on that, so. mrmissionctrl Mutants Wiki. Okay.
From Wikipedia, the free encyclopedia. Mycobacterium smegmatis. Mycobacterium smegmatis is an acid-fast bacterial species in the phylum Actinobacteria and the genus Mycobacterium. It is 3.0 to 5.0 m long with a bacillus shape and can be stained by Zeil-Nielsen method and the auramine-rhodamine fluorescent method. It was first reported in November 1884 by Lustgarten, who found a bacillus with the staining appearance of tubercle bacilli in syphilitic chunkers. Subsequent to this, Alvarez and Tavel found organisms similar to that described by Lustgarten, also in normal genital secretions, smegma. This organism was later named M. smegmatis. 1. Some species of the genus Mycobacterium have recently been renamed to Mycolicobacterium, so that M. smegmatis is now Mycolicobacterium smegmatis. 2. 3. Plaques from a virus isolated from a compost heap near Ukla. The bacterium is M.
Oh, what the hell? Well, that was enough of that right there to show you guys, I guess. Damn it. Boy, my computer's going slow. I'm a piece of shit, it ain't hard to fuck them down.
M. smegmatis is useful for the research analysis of other mycobacteria. M. smegmatis is generally considered a non-pathogenic microorganism. However, in some very rare cases, it may cause disease. 4. Use in research, edit. M. smegmatis is useful for the research analysis of other mycobacteria species in laboratory experiments. M. smegmatis is commonly used in work on the Mycobacterium genus due to it being a fast grower and non-pathogenic. M. smegmatis is a simple model that is easy to work with, i.e., with a fast doubling time, and only requires a biosafety level 1 laboratory. The time and heavy infrastructure needed to work with pathogenic species prompted researchers to use M. smegmatis as a model for mycobacterial species. M. smegmatis and M. tuberculosis and shares the same peculiar cell wall structure of M. tuberculosis and other mycobacterial species. 5. It is also capable of oxidizing carbon monoxide aerobically, as is M. tuberculosis. Edit. Smegmatis and M. tuberculosis and shares the same peculiar cell wall structure of M. tuberculosis and other mycobacterial species. 5. It is also capable of oxidizing carbon monoxide aerobically, as is M. tuberculosis. M. smegmatis is readily cultivatable in most synthetic or complex laboratory media, where it can form visible colonies in 3 to 5 days. These properties make it a very attractive model organism for M. tuberculosis and other mycobacterial pathogens. M. smegmatis MC2155 is also used for the cultivation of mycobacteriophage. Genetics and genomics, edit. The genomes of multiple strains of M. smegmatis have been sequenced by TIGA and other laboratories, including the wild-type MC2155 and some antibiotic-resistant strains before XR1R2.6. The genome of strain MC2155
pay attention, class.
9 mbp long and encodes ~6400 proteins 7, which is relatively large for bacteria. For comparison, the genome of E. coli encodes about 4,000 proteins.
4,000 proteins.
Shares more than 2,000 homologous genes with M. tuberculosis, and thus is a good model organism to study mycobacteria in general and the highly pathogenic M. tuberculosis in particular. The discovery of plasmids, phages, and mobile genetic elements has enabled the construction of dedicated gene inactivation and gene reporter systems. The M. smegmatis MC2155 strain is hypertransformable and is now the workhorse of mycobacterial genetics. Transformation, edit. Transformation is a process by which a bacterial cell takes up DNA that had been released by another cell into the surrounding medium, and then incorporates that DNA into its own genome by homologous recombination. See transformation of genetics. Strains of M. smegmatis that have particularly efficient DNA repair machinery, as indicated by their greater resistance to the DNA-damaging effects of agents such as UV and mitomycin C, prove to be the most capable of undergoing transformation. This suggests that transformation in M. smegmatis is a DNA repair process, presumably a recombinational repair process, as it is in other bacterial species. 9. Conjugation, edit. Conjugal DNA transfer in M. smegmatis requires stable and extended contact between a donor and a recipient strain, is DNase resistant, and the transferred DNA is incorporated into the recipient's chromosome by homologous recombination. However, in contrast to the well-known E. coli HFR conjugation system, in M. smegmatis all regions of the chromosome are transferred with comparable efficiencies, and mycobacterial conjugation is chromosome, rather than plasmid-based. Gray et al. 10. Reported substantial blending of the parental genomes resulting from conjugation, and referred to this blending as reminiscent of that seen in the meiotic products of sexual reproduction. See origin of sexual reproduction.
Keep that in mind.
DNA repair, edit. M. smegmatis relies on DNA repair pathways to resist DNA damage. Double-strand breaks are especially threatening to bacterial viability. M. smegmatis has 3 options for repairing double-strand breaks: homologous recombination, HR, non-homologous end joining, NHEJ, and single-strand annealing, SSA. 11. The HR pathway of M. smegmatis is the major determinant of resistance to ionizing radiation and oxidative DNA damage. This pathway involves exchange of information between a damaged chromosome and another homologous chromosome in the same cell. It depends on the Rec A protein that catalyzes strand exchange and the ADN protein that acts as a presynaptic nuclease. 11. HR is an accurate repair process and is the preferred pathway during logarithmic growth. 12. The NHEJ pathway for repairing double-strand breaks involves the rejoining of the broken ends. It does not depend on a second homologous chromosome. This pathway requires the Ku protein and a specialized polyfunctional ATP-dependent DNA ligase, ligase D. 13. NHEJ is efficient but inaccurate. Sealing of blunt DNA ends within a functional gene sequence occurs with a mutation frequency of about 50%. 13.
Remember that.
NHEJ is the preferred pathway during stationary phase, and it protects M. smegmatis against the harmful effects of desiccation. 12. SSA is employed as a repair pathway when a double-strand break arises between direct repeat sequences in DNA. SSA involves single-strand resection, annealing of the repeats, flap removal, gap filling, and ligation. In M. smegmatis, the SSA pathway depends on the REC-BCD helicase nuclease. 11.
That is Mycobacterium smegmatis, which, okay, so y'all following me? Still following me here, everybody? Everybody in? We got 8 people. Yeah, digital, just keep them busy. Keep them occupied. So, I was doling around the National Library of Medicine, right, for National Center for Biotechnology Information, because I was messing around with the IBM RxN chemistry, getting ready to run some chemical trials. I'll just flash you through some of these pages I was going over as I was working this morning. And I still can't get into this. They still haven't authorized me to get into this this search for the COVID-19 database. But here's the COVID-19 SARS-CoV-2 resources. And what I was going to do was like I showed you guys how to do, use the chemistry set here, the RoboRxN, the text to procedure, the atom mapping, the predict forward reaction, the predict retrosynthetic roots, and plant synthesis for your chemicals to make new drugs and and it's got the atom mapping. You can make DNA in this stuff, you know. So I went to PubMed or PubChem, and the last live stream I had went to substances and compounds, and I clicked on like that remdesivir is a popular substance, or let's try this COVID shield this time. So what I was doing was going down here and getting the code. Where is it at? Maybe it's not in substances. Maybe it's in compounds. I was looking at compounds. Yeah. And I went into the chloroquine. There's chloroquine right there. So I go into chloroquine, and there's your chemical structure and whatnot and everything. And so I wanted to have a base to go off of, right? There's the molecular formula right there. But here's the codes. And you want it's got all the information on it, all this information on these drugs, you know. And I'm not able to get into drug bank yet. They still haven't approved me. But there is just like a massive information about all these drugs. And it gives you the codes to plug into, where are they at? These are the ones, I believe. This one right here, the now called Smiles, the Smiles. I believe this is it. You get this code, you get the Smiles, the canonical Smiles code. You copy it, you run it over here to the RX mapper, you get the source code. No. That's not the source. You get the fucking demo. The demo. There we go. And you go. You just delete this right here, and then you. So anyway, this is what I was doing to map the atoms, right? With some bad reaction. So I added it. What I did was I added it to this. It still didn't work. It mapped the Smiles. It mapped the Smiles. Mapping. I don't understand. It worked for me last time. Maybe I don't have the right code. There, it's the Dale's Adler. It's the Dale's Adler code is the Smiles code, I think. Anyway, it gave me it was allowed me to add that to the example, and so I figured that I could just plug it in, right? Plug in the but it doesn't let me. So what's the example? Anyway, we'll get down to it here in a second. I just wanted to go over this real quick, what I was doing. Oh, maybe it was missing an equals. Maybe it was missing an equals. I have to put equals. Maybe. I don't know. Fucking, I don't know anything about fucking this shit. I'm just kind of just no, that didn't work. I don't know. We'll have to figure that out later. But I'll get to figuring out how to run this thing. But anyway, you map the atoms, and then you get this. You copy that over here. You take it to your RX bin, and it's still not letting me do it. Maybe if I go to a maybe if I add two of them is what it is. Anyway, if this doesn't work, fuck it. But I thought I understood how this worked. That says bad smiles map. Yeah, that didn't work either. I don't know. It works when you add it on to one of the things. I don't know how to write fucking this shit. So you know, it really, I have to go read a book. Anyway, I was looking at that, and I came across this part here, right? So I was okay. So I don't want to look at the nucleotoids, and I don't want to look at the proteins. Are you guys still with me? Some people, a couple people dropped out. That's okay. That's okay because you know we're gonna be here for a while. So I didn't. So I looked at genomes, and I was like, well, there's the genome, and there's 38 proteins and one nucleotide. Same here, same thing here. It takes you to the same page. If you were here and you chose one of these, the genome, protein, nucleotides. So I went with the RefSec proteins, and then it has the RefSec genome. It's all the same stuff, right? Okay, you following me? So those pages were the same, even though it looks like you could choose different ones. So I went to the proteins, to the 38 proteins, and I was just looking around at what kind of proteins these things had, and I was like, well, that doesn't tell me much. So I scooted it over, and then it has the names here: leader protein, and one of these proteins looked not like the other. And I see this methyl transferase. And I was like, Ferase? That sounds funny. I'm going to check it out. Okay? So I click this, and it gives me this little panel. Right? So I click this, and the 4 publications and the 1 project. I open those up. That's the complete write-up for it right there almost. Severe acute respiratory syndrome coronavirus 2. Organism. And then I go over here and look at this. And it's got some publications, a new coronavirus associated with human respiratory disease in China. And I was just thinking, okay, big deal. I get to looking and get over here to this. And I'm looking at it, says multi-species. And I'm like, that's strange. So I hit the biosample. I hit the PubMed and the biosample. And I start reading this PubMed links for ID 485481. A novel phage infecting Alzheimer's represents a distant group of sycophages. Genome sequence organism of the Mytha family, persuaded the LTI. And I'm like, what is that? That sounds like CRISPR to me. While genome sequence analysis, forcing blah blah, all this shit, right? And then this one I came across is the one we're looking for. A viral sample from Mycobacterium phage. Complete genome sequences of these phages. So I'm like, that's CRISPR stuff. So I check out this. I was like, what's this weirdo 19ES? Is a novel singleton mycobacteriophage, and that's when I searched the mycobacteriophage. I mean, that's when I searched the M. smegmatis mutans and found out it was a macrobacteriophage. So Weirdo 19ES is a novel singleton mycobacteriophage that selects for glycolipid deficient phage resistant M. smegmatis mutants. So that is our Weirdo 19 does not exist in the Wikipedia. that is off of the one project where I got this from. It was from this project, and I went to look at this, the publications. A novel phage infecting as a group of sickle phages, and I was like, that's fucking CRISPR, isn't it? And it's got all the nucleotides, protein sequences. And I just remember that one that name stood out, that weirdo, because I was like, that has to be something to do with the three-legged thing that the French guy found, right? So I'm trying to make heads or tails of all this, and so we'll read this here. Y'all still with me? What's chat say? The context is blurry. Probably because I got. Let's see if I open a tab. Now, can you see it now? Yeah, you can see it better now, can't you? Is that better? I mean, I want to make sure you guys can see it. Okay, so we're going to start reading this.
WATCHDAWGS is a novel singleton mycobacteriophage that selects for glycolipid-deficient phage-resistant M. smegmatis mutants. Christian Alejandro Suarez 1, Jorge Lina Judith Francescella 1, Sabrina Emil Sticella 1, Hector Ricardo Morbidona 1. Affiliations. Collapse.
Now,
Affiliation.
Now hold on. Let me get point this out to you. This is all found in the proteins, okay, the 38 proteins that are in the SARS-CoV-2 data hub of being the nucleotides, the proteins. Let me show. I didn't point this out. I want to make sure the context is right here. Okay, I have to make sure the context is right, or we're not going to be seeing eye to eye through the rest of this whole thing. What is this? NCBI virus. Okay, now you see this. Y'all see this right here. Refine results. This says SARS-CoV-2 data hub. Download. Download what? Data type. I want the protein list. Download all records. Boom. Boom. Weirdo. I got the weirdo bacteriophage JPEG right there. I got a bunch of information about this weirdo thing. So the beta coronavirus blast, the blast feature is pretty cool. The outbreak information is old from last year. It's got articles in PubMed. It's got NCBI SARS-CoV-2 resources, data sets, SRA data. Okay, so this is the SARS-CoV-2 data hub, the dashboard visualizations, mutations in SRA, and the complete tree of the biological information of SARS, severe acute respiratory syndrome, coronavirus 2, SARS-CoV-2, taxid, the tax ID 2697049. Okay, so it was looking for the virus. This is the virus. Okay, and this is what's in the virus. See if I close this out, it just starts looking at all the entire the ref sequence. So you can search proteins. I can go. That's not right. What if I do it like this? Oh, how they add it up there? Explore virus data. So I have to go back. SARS-CoV-2, like that. Go down to the proteins. No suggestions found. Oh well. Anyway, I don't know how to work this shit, really. I just know that I went over here and looked at the proteins because there was 38 of them. There's one nucleotide, severe acute respiratory syndrome related coronavirus, isolate Wuhan lab, and the genome, right? China, coronavirus. So this is coming from the, there's all the protein ascensions and the nucleotide ascensions. So that YP, what is it? God. Losing myself here. See, I'm trying to just link it all up, but YP009725311101 is the NCBI reference sequence. Genome details. Well, we're looking for the proteins. And this one, there it is, 009725311. It's that ribose. So I just thought, I was just kind of fucking around, and I come across this. I thought that was interesting because, I had meth in it, had the word meth in it. So I clicked on it, and it just blew my mind what it turned out to be. So back to work here. Yeah, this is the one with the pictures. We're going to read some of this one. And
G-Matus mutants. Christian Alejandro Suarez. WATCHDAWGS is a novel singleton mycobacteriophage that selects for glycolipid-deficient phage-resistant M. smegmatis mutants. The sequencing and bioinformatics analysis of bacteriophages infecting mycobacteria has yielded a large amount of information on their evolution, including that on their environmental propagation, on other genera such as Gordonia, closely related to Mycobacterium. However, little is known on mycobacteriophages' cell biology, such as the nature of their receptors or their replication cycle. As part of our ongoing screening for novel mycobacteriophages, we herein report the isolation and genome bioanalysis of WATCHDAWGS, a singleton cipher-virity temperate mycobacteriophage with a 70.19% GC content. Nucleotide and protein sequence comparison to actinobacteriophage databases revealed that WATCHDAWGS shows low homology to Gordonia phage ruthie and mycobacteriophages falling in clusters Q and G, and to singleton DS6A. WATCHDAWGS also displays uncommon features such as a very short lysine A gene, with only one enzymatic domain, and two putative HNH endonucleases. Mycobacterium smegmatis. Mutants resistant to WATCHDAWGS are cross-resistant to I3. In agreement with that phenotype, analysis of cell envelope of those mutants showed that WATCHDAWGS shares receptors with the transducing mycobacteriophage I3. The singleton mycobacteriophage adds up to the uncommonness of local mycobacteriophages previously isolated by our group and helps understanding the nature of mycobacteriophage receptors. Figures. Citation: Suarez C.A., Franciscella J.J., Tiselli S.E., Morbidoni H.R., 2020. WATCHDAWGS is a novel singleton mycobacteriophage that selects for glycolipid-deficient phage-resistant M. smegmatis mutants. Helo S115, 5E0. Bacteriophages infecting the genus Mycobacterium are to this day the best characterized bacterial viruses, with more than 1,700 genomes sequenced and roughly 12,000 phages isolated. Several reports have described the development of the bioinformatics programs needed for mycobacteriophage analysis, as well as the construction of mycobacteriophage-based tools made for genetic manipulation of mycobacteria, such as the recombining method to modify mycobacterial and mycobacteriophage genomes 1, 2. Taken collectively, the information produced through genome sequence analysis gives an ample view of the scenario of mycobacteriophage evolution. As an example, the characterization of mycobacteriophage patients revealed a low GC content, 53%, and codon usage that did not reflect those of its laboratory host, Mycobacterium smegmatis, but was more related to Mycobacterium abscessus 3. Similarly, phages infecting Gordonia species showed homology to mycobacteriophages, suggesting propagation across genera.
Remember that.
Several mycobacteriophages were isolated and characterized by our group, with the aim of exploring their diversity at a geographical location, South America, in which little work on that matter had been carried out 5 to 7. Mycobacteriophages defining novel clusters and subclusters were detected by our group in spite of the reduced number of total isolates, less than 38. One of such phages, Weirdo 19ES, initially named 19ES, was isolated in Argentina from soil samples during a previous screening. Preliminary characterization by TEM of Weirdo 19ES showed that it has a cipher-viridae morphology with a length of approximately 250 nanometers and an icosahedral capsid of 70 nanometers. This phage produced small turbid plaques at 30 degrees Celsius and larger plaques at 37 degrees Celsius, but failed to propagate at 42 degrees Celsius, behaved as temperate, and failed to infect M. tuberculosis of 5. Surprisingly, Weirdo 19ES propagated in M. smegmatis. Without addition of extra calcium magnesium cations to the growth medium, a common requirement for most mycobacteriophages. 5. Preliminary analysis of Weirdo 19ES genome sequence indicated that it was a singleton, Francescelli, J.J., personal communication. In sum, those features of this mycobacteriophage led us to pursue its full bioinformatics analysis, the characterization of its life cycle, and nature of its mycobacterial receptors, results that are reported herein. Materials and methods. Strains, chemicals, culture media, and growth conditions. Mycobacterium smegmatis MC2155, lab stock, and derivatives were grown at 37 degrees Celsius in Middlebrook 7H9 broth supplemented with 0.5%, with V, glycerol, 10% ADS, albumin dextrose NaCl, and 0.2% with V, tween 80 hereafter designated as 7H9 ADS GLYTW for short, as liquid culture medium. The same medium devoid of tween 80 and supplemented with agar 1.5%, with V, was a solid medium, 7H9 ADS GLY agar. Mutant strain M, smegmatis MSMEG_0398, TN61, Mic55, a glycopeptidolipid, GPL, deficient mutant generated through insertional transposon mutagenesis, was the generous gift of Dr. M. DAF Institut de Pharmacologie, et de Biologie Structurale, Toulouse. Mycobacteriophages I3 and D29, used as controls throughout this study, were lab stocks. All chemicals and solvents were purchased from Sigma, St. Louis, Mo, unless otherwise stated. General bacteriophage techniques. Preparation of mycobacteriophage high titer lysates and their titer determination were done as previously published. 5. Briefly, late log phase culture of M. smegmatis MC2155 grown at 37 degrees Celsius was spun down and washed twice in phage.
Okay, we don't need to know all that. When it gets down here, molecular biology techniques, chromosomal DNA extraction, and derived strains are performed according carried out using primers, blah, 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 bioinformatics analysis. Well, we get down here to Weirdo 19 genome organization, analysis of the left side of the genome of Weirdo 19, the central region, integration and immunity region, comparative genomics, and I get down here and I see this right here. This is very important.
There are very few reports on the biology behind M. smegmatis resistance to mycobacteriophages, or the nature of receptors for mycobacteriophages, being the mutants resistant to the transducing phage I3 the best characterized till now 43, 44. In order to gain knowledge on the subject we obtained several spontaneous M. smegmatis mutants resistant to Weirdo 19ES, see materials and methods, one of which, designated 1R Weirdo 19ES, was kept for further analysis. Simultaneously, we isolated spontaneous M. smegmatis mutants resistant to mycobacteriophage I3 following the same protocols, keeping one clone, which we designated as 3RI3. Importantly, successive passages on fresh medium showed that morphotypes were stable although revertants, with colony morphology similar to the parental strain arose with a frequency of 10, 6,
morphotypes. Okay, remember that. Morphotypes.
In order to characterize those phage-resistant mutants, aliquots of each one of them, along with the parental strain, were plated onto solid medium in the presence of the hydrophobic dicongo red. Observation of the plates at low magnification revealed that I3 and Weirdo 19ES Smegmatis-resistant mutants displayed a rough colony phenotype, being approximately half the size of that of the wild-type M. Smegmatis MC2155, Fig 9 Panel A. M. Smegmatis Mike 55, a TN611 transposon insertional mutant affecting GPL synthesis 45 that was added as control, also displayed a highly similar colony morphology in agreement with what was described in the literature. Fig 9 Panel A. The behavior of the phage-resistant mutants on sliding motility was also tested, finding that while the parental strain displayed the well-known growth pattern, the three mutants, 1R Weirdo 19ES, 3RI3, and Mike 55, failed to move from the point where the cultures were spotted onto the plate. Fig 9 Panel B.
Now,
in concordance with the loss
where
of the
have we
sliding
seen
motility,
that
the
before,
aggregation index
guys?
showed that the three mutants were much more prone to
What kind
aggregation
of three-legged,
five-fold than the wild-type strain of Fig 9.
3 or 4-legged alien
Download.
creature they were talking
PPT.
about,
PowerPoint
right?
slide. PNG. Larger image. TIFF. Original image. Fig 9. Phage-resistant mutants of Mycobacterium smegmatis. The mutant strains, 1R Weirdo 19ES, resistant to Mycobacteriophage Weirdo 19ES, 3RI3, resistant to Mycobacteriophage I3, or Myc55 share colony morphology, Panel A, sliding motility behavior, Panel B, and cell aggregation, Panel C. Panel A, aliquots containing 102 CFU of each strain was spread D onto solid medium plates containing Congo red, 100 gml1. Growth was examined under microscope at 8x magnification after 3 days of incubation at 37 degrees Celsius. Pictures are representative of 10 fields observed for each plate. Panel B. 5L drops of a stationary culture of each strain were placed in the center of 0.3% with V. agar plates. The plates were sealed with parafilm and incubated for 7 days at 37 degrees Celsius. Panel C. Aggregation index for each strain was determined in the absence or presence of 0.5% WW, tween 80 as described in materials and methods. https:// colony morphology of 1R Weirdo 19ES and 3RI3M, smegmatis-resistant mutants suggested cell envelope alterations. Hypothesis supported by the colony size and morphology shared by these two mutants, and Mike 55. A previous report on transposon insertions causing resistance to I3 and yielding a rough colony phenotype and loss of GPL synthesis led us to extract and analyze cell envelope lipids of 1R Weirdo 19ES, as described in materials and methods 1245. Extraction and chromatographic analysis of lipids of the cell envelope of M. Smegmatis MC2155, 1R Weirdo 19ES, 3RI3, and Mike 55 supported our hypothesis, as no polar GPLs were discernible by TLC analysis in the three mutants, while the parental strain displayed the normal content of polar and apolar lipids.
Okay, the lipids, right? Now, I know some of you guys aren't following this, as well as others, and I get that. But the whole point here is there is CRISPR bacteriophages that they had fucked around with right there. That was the proof that they were taking. They would alter the DNA, the structure of these bacteriophages they found in the rainforest there in the mud, and somehow it wound up in the sequencing of the SARS-CoV-2. Is my point. Now, do you follow? Leslie's following me. Digital, are you following me? Frigga, I explained this all to Frigga, and Frigga probably went to bed by now. Frigga, you still here? White boy, you still here? You following me? You get what I'm saying? There's CRISPR technology in the fucking SARS-CoV-2 virus to begin with. These are samples from China that they're dealing with in the NIH lab. Okay? So this proves that they were not only gain of functioning, but they stuck CRISPR fucking shit into the virus. bacteriophages that can alter your DNA, that are doing totally something different than what the spike proteins or the functionality of the mRNA spike proteins and how our cells are going through this process of producing the spike proteins to ward off the big bad coronavirus. When, from what I understand, the spike proteins are the fucking coronavirus, or these bacteriophages are the coronavirus, right? So this takes a whole new outlook bacteriophage, xanthophage from biosample. They're all from the fucking Wuhan, severe acute respiratory. All these geolocation, China. Okay, from the SARS-CoV-2 data hub. I'm not giving out fucking false information. This is coming from the NIH, guys. 2019-12. December of 2019, they found this. 2,0-ribose methyltransferase. They found this in there. They found it to be a fucking one project had been done on it. It's a novel phage infecting Altroromus, represents a distant group of sycophages infecting diverse aquatic Cophiotrophs, right? Was that shellfish? Look at the 5,000 publications about it. Salmonella, papillo. It has. They've engineered it to fucking go with anything. So much fucking R&D into this stuff, dude. And it was talking about echinaceas, and it could have said something about hydras too. I don't know. Data sets, publications. PubMed's got 5,000. PNC's got 2,000. Protein sequencing. Genomic DNA. Genomic RNA. This was found in SARS-CoV-2. Look at all the projects they've got going on with it. See? Organism, right there. Severe acute respiratory syndrome, coronavirus 2. Complete genome. SARS-CoV-2, human immunodeficiency virus. Hepatitis, all these projects related to that. Zika, dengue, measles, Middle East respiratory syndrome. The whole fucking all the assemblies. They've got the genomes for all this shit, man. Thai forest Ebola virus. What's that? Sudan Ebola virus. Equine arteritis viruses. I wonder what's at the fucking last. Some shit we never heard of, right? Tulip mild model mosaic viruses. Dicasimophora. We can't even say that. Salmonid herpes virus. That's the last one. I bet that's a fucking humdinger. Wisconsin epidemic. I'm just looking around here right now. But this publication had it good. See that? That looks like that entity that they said had 3 legs and it looked like a hydra, right? And all those things, they said it could have been nano. But they're not. They're bacteriophages. And it's called this is figure 9. Refer to this as this is the paragraph I wanted to read. Biology cycle of weirdo 19 ES. Because this is the one that's the star of the show here.
Biology cycle of weirdo 19 ES. To add information of mycobacteriophage weirdo 19 ES, we determined some of its biological features, such as infection cycle parameters and phage bacterial interactions. The C materials and methods. Our analysis showed that weirdo 19 ES was able to lysogenize the mycobacterial host at a frequency of 12%, an average of 3 independent experiments, which is intermediate between the frequency displayed by L5 per 22%, or Giles 2%, as previously reported too. This singleton also had an unusual lytic cycle characterized by an extremely long latency time of almost equal to 300 minutes, which largely exceeds those reported for other mycobacteriophages, such as Campi, which showed a latency time of 100 minutes 40, or D29, 100 to 120 minutes, 41, 42. There are very few reports on the biology behind M. smegmatis resistance to mycobacteriophages, or the nature of receptors for mycobacteriophages, being the mutants resistant to the transducing phage I3 the best characterized till now 43, 44. In order to gain knowledge on the subject, we obtained several spontaneous M. smegmatis mutants resistant to weirdo 19 ES, C materials and methods, one of which, designated 1R weirdo 19 ES, was kept for further analysis. Simultaneously, we isolated spontaneous M. smegmatis mutants resistant to mycobacteriophage I3 following the same protocols, keeping one clone, which we designated as 3RI3. Importantly, successive passages on fresh medium showed that morphotypes were stable, although revertants, with colony morphology similar to the parental strain, arose with a frequency of 10-6-10-7. In order to characterize those phage-resistant mutants, aliquots of each one of them, along with the parental strain, were plated onto solid medium in the presence of the hydrophobic dicongo red. Observation of the plates at low magnification revealed that I3 and weirdo 19 ES. M. smegmatis resistant mutants displayed a rough colony phenotype, being approximately half the size of that of the wild-type M. smegmatis MC2155, Fig 9 Panel A. M. smegmatis Mike 55, a TN611 transposon insertional mutant affecting GPL synthesis 45, that was added as control, also displayed a highly similar colony morphology in agreement with what was described in the literature, Fig 9 Panel A. The behavior of the phage-resistant mutants on sliding motility was also tested, finding that while the parental strain displayed the well-known growth pattern, the 3 mutants, 1R weirdo 19 ES, 3RI3 and Mike 55, failed to move from the point where the cultures were spotted onto the plate, Fig 9 Panel B. In concordance with the loss of the sliding motility, the aggregation index showed that the 3 mutants were much more prone to aggregation, 4 to 5-fold, than the wild-type strain, Fig 9. Open in a separate window. Fig 9. Phage-resistant mutants of Mycobacterium smegmatis. The mutant strains, 1R weirdo 19 ES, resistant to Mycobacteriophage weirdo 19 ES, 3RI3, resistant to mycobacteriophage I3, or Myc55 share colony morphology, panel A, sliding motility behavior, panel B, and cell aggregation, panel C. Panel A, aliquots containing 102 CFU of each.
So this panel B here is showing it's basically its legs. It's sliding motility behavior. And I Wikipedia that. And where is it at? Sliding motility behavior. Gliding motility. And it says
mechanisms of this motility. Gliding motility is a type of translocation used by microorganisms that is independent of propulsive structures such as flagella, pili, and fimbriae. Wand gliding allows microorganisms to travel along the surface of low aqueous films. The mechanisms of this motility are only partially known. Twitching motility also allows microorganisms to travel along a surface, but this type of movement is jerky and uses pili as its means of transport. Bacterial gliding is a type of gliding motility that can also use pili for propulsion. The speed of gliding varies between organisms, and the reversal of direction is seemingly regulated by some sort of internal clock. For example, the apicomplexans are able to travel at fast rates between 1 to 10 ms. In contrast, Mysococcus xanthus bacteria glide at a rate of 5 m a minute, 3-4. Cell invasion and gliding motility have trap, thrombospondin-related anonymous protein, a surface protein, as a common molecular basis that is both essential for infection and locomotion of the invasive apicomplexan parasite. 5 micronemes are secretory organelles on the apical surface of the apicomplexans used for gliding motility.
so bacterial gliding. Bacteria moves under its own mobility, right? So doesn't that look like the feared alien creature we were all so freaking out about that they found? Though it's a hydra, it's not a hydra. It's a bacteriophage. It's got legs. It's a little organism. So I just wanted to bring that point home. I still haven't figured out this RX mapper yet. I think I'll save the CERN for tomorrow night. I'll give you a taste of what it is, because I mean, there's nothing we can do about it. We're subject to all this stuff, man. I mean, whether you think it's aliens or, like Leslie says, the dark soul trap matrix, the dark ones, you can't see them, but they're controlling everything, right? From where? I don't know. From their little micro nano houses inside of us is where they're controlling it from, Leslie. They're not big shadow entities the size of mountains floating around in space. They're little bitty fucking microorganisms floating around in us and in the mud in the rainforest and shit. These motherfuckers can communicate with each other's colonies of them, billions of them. They outnumber us. Billions and billions and trillions, quadruple trillions, all throughout the fucking planet. You get what I'm saying? When they say these things that we're seeing have been here longer than we have, they're absolutely right. They have. They came from the center of the planet. That's where you get your oil producing blue algae from the bottom of the ocean and stuff. And all that stuff propagated from the interior of the planet. These planetoids, these stars, they are life producers. And I don't think everybody hasn't grasped that yet. You know, when we go back to back to ground, back to Earth, we truly do. We've got all these organisms. Each cell can be really classified as a different life form in our bodies. Each bacteria, the cells, and the white blood cells, the lichens, all the different operations, all the different types of organisms that are in your body that make up who you are, and you're like, why isn't there a cacophony of voices and some people do have a cacophony of voices in their head, and next time you think of where that voice in your head's coming from, it could be coming from like your lower intestine, Mr. Bacteria down there telling you something. These things are conscious, dude. They got consciousness to them. The basic structure of consciousness of the universe. There's a collective consciousness that makes up the universe, and we're not apparently in control of it, where it's going. I think these things are. Okay? That guy, that red magician back there, he's pointing to that star for a reason. It's because that's where life comes from. The center of stars, the center of planets, and exudes out from microorganisms, from the basic. And these things have consciousness, and they're the aliens, guys. They're the three-legged fucking. They're us. They're us. But now I'm not going to go into the nanoparticles or anything. But I do want to show you this, what I did find. And we'll hit the gematria on this real quick. So I was looking at the my eyes can't take this fucking light. I was looking at the geomap, okay? And it only gives you data to back to December of 2020. Okay? So I was like trying to get more information. And I went up here to the About section. And I hit this white shirt group, which I'm going to have to change my screen share again. This white shirt group. Now, how do you say it? Frigga, you say it. It's not white Wiseheart. It's white Wizchart or something. It's German. Anyway. This group. Wizchart Research Group. This is the guy. This is what was running the COVID database information for the NIH. Okay? Check this
Welcome
out.
to the Wishart Research Group homepage. Dr. David Wishart, PhD, Yale, 1991, is a professor in the departments of Biological Sciences and Computing Science at the University of Alberta. He is also a senior research officer and the director of the Nano Life Science program at the NRC's National Institute for Nanotechnology, NINT. He has been with the University of Alberta since 1995.
Nanotechnology. These guys have been in bed with this all from the very beginning.
His lab's most significant contributions have been in the area of protein chemical shift analysis and the prediction. He has been with the University of Alberta since 1995. Dr. Wishart has active research programs in structural biology, nanobiology, synthetic biology, prion biology, bioinformatics, and metabolomics. Some of his lab's most significant...
Prion. Now that's what you get when you eat brains or human flesh. You get prion disease.
Significant contributions have been in the area of protein chemical shift analysis and the prediction of protein structure. Dr. Wishart has directed a number of core labs at the U of A over the years, including the Faculty of Pharmacy's Mass Spectrometry Core from 1998 to 2002, the Polyoncs Bioinformatics Core, 2001 to 2004, the Pence Bioinformatics Core Facility, 2002 to 2005, and the Genome Canada Bioinformatics Help Desk, 2003 to 2011. From 2006 to 2009, Dr. Wishart led the Human Metabolome Project, HMP, a multi-university, multi-investigator project that catalogued all of the known metabolites in human tissues and biofluids.
You got that?
Using advanced methods in NMR spectroscopy, mass spectrometry, multidimensional chromatography, and machine learning, Dr. Wishart and his colleagues identified or found evidence for more than 8,000 endogenous metabolites. This information has been archived on a freely accessible web resource called the Human Metabolome Database. The methods and ideas developed for the HMP have helped lay the foundation for a number of other metabolomic databases, DrugBank, T3DB, FoodDB, and metabolomic software tools, MSEA, MetaboAnalyst, MetPA, MetaboMiner. They have also led to the development of a number of interesting clinical metabolomics projects and collaborations. These include studies of several cancer biomarkers, the identification of organ transplant biomarkers, and exploring wound healing mechanisms. The Wishart Research Group is located in Edmonton, Alberta, Canada. Please visit E. 1991, is a professor in the departments of Biological Sciences and Computing Science at the University of Alberta. He is also a senior research officer and the director of the Nano Life Science program at the NRC's National Institute for Nanotechnology, NINT. He has been with the University of Alberta since 1995. Dr. Wishart has active research programs in structural biology, nanobiology, synthetic biology, prion biology, bioinformatics, and metabolomics. Some of his lab's most significant contributions have been in the area of protein chemical shift. Welcome to the Wishart Research Group homepage. Dr. David Wishart, PhD, Yale, 1991, is a professor in the departments of Biological Sciences and Computing Science at the University of Alberta. He is also a senior research officer and the director of the Nano Life Science program at the NRC's National Institute for Nanotechnology, NINT. He has been with the University of Alberta since 1995. Dr. Wishart has active research programs in structural biology, nanobiology, synthetic biology, prion biology, bioinformatics, and metabolomics. Some of his lab's most significant contributions have been in the area of protein chemical shift analysis and the prediction of protein structure. More about our group. Copyright symbol Wishart Research Group 1996, 2021. Hybrid nanoelectronics. Collectively, advances in hybrid nanoelectronics has great potential to have a profound impact on global computing architectures and electronics fabrication, leading to added value in a variety of industries. We are active in seeking partners to deploy the latest advances in this area. Copyright 2019 Mint in T. View rude words.
Dang it. Okay, so the gematria for Weirdo 19ES is Jewish is 1143, English is 588, and simple is 98. Okay. Weirdo 19ES is not in our gematria database. Okay? I'm the first one to search it. Add it. It's added now. It's officially gematria. All right, guys. We're going to tweet this bitch out. Deep State Watchdogs. Oh, fucking John B. Wells, you ain't got nothing on me. You can't match this, is right. Stupid XRP Discord. Wormhole. This, I'm just going to scroll through this, and I looked at it earlier, and it was pretty amazing. December 10, 5, 6, December 16th. To be calm is good advice. An unforgettable year. Serpent Void. For justice stuck on you. Swollen head. Hartford Distributors. Sanders murder lead. Problem bears tragically hip. Unforeseeable intercept. Never said that. Like it. Keep wishing. November the death. Jennifer Harrison. Okay, here it is in English. Ray is a deck ID. God reigns. First God. God himself. Big Bang Light. I am Lord. Yes, Lord. Tom Brady. Trust. Grammatical. Dragon Inky. Crystal. Now this is the 588 and the 98. Right? The English and the simple matches. Chosen one. Anything. Beat, Deck, Zodiac, Prophet, God is Karma, Has Proof, God is Hidden, Apple Tree, God is Faith, Garden of Eden, Democrats, I Am What I Am, the Serpent, Black People, Nero Caesar, Lazarus, Istanbul, Trevor, Seeger, Flowers, Angel of Death, Contagion, So True, Python. So load more results. It gets interesting on the second or third page. What are you doing, Tina? Germany cipher runes. Extremely tired. Anxiety cure. Who is Capula? 1960. Epstein is alive. Aquarius Pisces rising. AB positive blood. 5044. You are the sea beast. Angel wing begonia. River of fire. Complete psychopaths. Wide pigskin. I saw them. Diane Downs. You are protected as he wills from the volcano. Invisible pass. Apocalyptic parasites. Apocalyptic parasites. Okay. Mark White. I'm just reading some of these. I'm not reading all of them. British Houses of Parliament. Christopher Jaden. Mapping synchronicities. Remember me talking about that? Frigga and everybody. Hated Anthony Bourdain. From Below. To see how this goes along with what's going on? Karl Marx, Valentini, Anxiety, Elevator, Dead Cat Bounce, Black is King, Do Not Fear, Blue Angels, Israelite, O Negative, Mission, You and Me, Independence, Deliverance, Luciferian, James Harden, Pure Gold, Waterfall, Rosary, Have a Nice Day, I Need You, Olivia Ann, Blizzard, Proton, Beam of Light, Golden Key, Zephyr, Montreal, Oblivion, the 3-5, Find Me a Girl, Tyrant, Snowball, Marquis, like Florida Marquis. Got an interesting gematria, doesn't it? Lie August of Russian. The abominable treatment of the poor. Okay, look at this, how it's going. Ambrosia Chocolate Factory. Fractals of Tigers rising up. And these are the matches, searches, 106 searches for each one of these. On the very far right, it's the searches. And then it's Jewish English simple searches. So the Jewish matches are 1143. Life secret revealed. Use lots and lots of lube. Fear weakens. Gematria overlord. An atrocious attitude. The spread of evil. This is my mission in life. Anthony Kennedy, Broken Wing. Do you sense that? All the parasites are getting deleted. Pythagoras Pressures, dead at 68. John Stossel. Gone till November. Gone till November. Who's gone till November, guys? Maybe you're Jesus gone till November. So, or the devil, who knows? The entities or whatever are gone till November. Moon month menstruation. Here's the English, it's simple. This is interesting too. Blue star. Frigga. What have we been looking at for moving around in the western sky? That blue star. And also, Venus looks blue right now. Carbon tax. King George. Dr. Pepper. Lung cancer. Me and you. Snickers. All this stuff rings a bell to me. Black Knight, Get Lost, somebody. Day of the Dead was just the other day. Goodness, 9-3. Station. Romanov. If I would have kept going on Halloween, doing what I was doing, I would have come across all this stuff. It would have been the Day of the Dead. Thanatos, County, Sentinel, Rosetta, Ping Pong. Second Death. What can I do? Tarsus. You know, Tarsus, the city of Tarsus. The Prince. The Eighth King. April 9, Lockwood. Yes, to get her. Champions to know. Amanda Knox. Like Amanda Knox Bind. Cherubims. Holy Bride. And we're only getting down to the searches of 70, 68, and then the other ones, the English and Simple have 700 more to go, but we're getting down to the 50s and 60s into the Jewish search results. 35th degree Mason, astronomical alignment best tool. Greys are our allies. Satanic ritual murderers. We'll meet again. A nation led by serpents. Seagulls killed by lightning. May 6th and mischief. Irresponsible Hugh Monster. Remember me talking about monsters earlier, Frigga? Human monsters? Money came to me in abundance. Not me. Maybe somebody here. Arrhythmus has no human soul. My right side is stinging. Oh, that was me earlier today. The Christian apocalypse. Justice Serial. From the end of heaven. Psycho gang stalking liars. Japanese Anunnaki. Exposed lying females. All love is in the air. Increasingly delusional. It is my duck face Markbot. Ooh, everybody knows duck. Everybody knows deplorable duck. And Akam Shabara, they are calling me insane. Being a ladies' man ain't cool anymore. Peter DeVries, the Eternal Crime Syndicate. Obama forces the mark of the beast on mankind. Magnificent of the holy couple. Minesweep. No humans belong in Saturn. Biden is Sanada, the evil. Plasma wall. Politicians are a joke. Biden is Sanada. Who is Sanada? We'll have to check that out. That's definitely Biden is Sanada. Let's go. Let's take a. You see how this is going. We'll blindness, cluster, morphine, Rupert, somehow, 5-3, Michael John. And if you keep on going through the search results and get lower and lower, the more it tells a story, the more it makes sense. God first. White magic, Eye of the Nine, Transcend, Hand of Fatima, Magic 7. Now this is as far as I went earlier. So this will all be stuff I haven't seen yet. ChristRapture.com. Well, I've seen some of this. 304 Blame Room. Queen of Transformations. Male, female, joy and holiness. Thus Lucifer, not Satan. Huge announcement coming soon. Goats are smarter than people. Fake sons of Zeus. The Anunnaki Enki, father of humans. I cancel it in November. The Dutch system. Or the Deutsch system. The false deliverer. Irving Santiago. A major death, December 8th. And then remember that other date, December 16th? Was it? Yeah, 16th. Mocking and ridicule, December 8th. And the 16th. Designated driver. Please destroy Gematria. Hijacking evangelical preachers. Serpent-possessed human. serpent-possessed human. My eye cut. Latin beats the eye, the symbolism, the eye symbolism. Truth bombs, not real ones. Fancy shatter entire matrix. See what we're doing here? Humanism. Humanism, not transhumanism, but humanism. Okay? I am Groot. 8801. Bilderberg. Vlad Dracul. All his lies. Nice work. Barack's wife. Tuttle. 11 cubed. See how it's talking to me? Aid of deity. Adonai Adonai Malikith. That is Joshua Olphus. Adonai. Adonai Malik. I'm going to write that down because I need to see exactly who that is. Pleiadian race. Arur. Khalif al-Baghdadi. Khalif al-Baghdadi. Who's that, Frigga? Ukrainian. Rob Keller, all for one. I think I can. I feel you. I get that. I get people telling me I feel me all the time. Allison, Curtis, Walters, Cold Snake, Buzzard, Critics. Now this is the Weirdo 19ES in Gematria. No longer desires fame and fortune. 711, self-centered psychopath. Our Lord is you Christi. Serpentine Deluge. Man is forgiven. Amen. Central Illusion Agency. Yahweh's Chosen Pleiadians. Spiritual Book of Discernment. Baphomet, Phallus, Mark of the Beast. Ray Archetype. Finite number of statements. Lucifer's Antichrist. I think the gematria on this is more revealing than anything, really. Black Sun, Cube of Saturn. A Christ, Deck 7. You make me sick and sad in both. Ophi David of Ethiopia. That Ophi David of Ethiopia. That could be my buddy from Ethiopia there. Magnetic Geometries of Saturn. Brotherhood of Luxor. Mad Turks from Istanbul. Destroy all bad remains good. Decode your script. Bunkers of Pike Creek. Reclamation of Christos mission. And at 36 search results for 36 searches, and it hadn't been added for Weirdo 1986. So I'm the 36th person to search this. With some of these things having millions of search results, searches involved with them, like Barack Obama and stuff like that. Zim's reclamation of Christos mission of December. There it is. That's close to the search result. December 16th. I think that's right. 10 or the 4. That's December 14th. And the 14th. I thought I'd seen the 16th somewhere. Earthquakes and pestilence is right close to where the search results for weirdos is. Earthquakes and pestilence. Audio, video, RAM. My desire, great beast, selenium. Cosmic law, ungodly, even now. Cremation, seed of Abraham. Pluton, shellfish. Shellfish is where they are getting these fucking CRISPR, these bacteriophages. I believe, shellfish, decomposing shellfish. Boondocks. Be content. Jamie Bergman says, yeah, I've been being content for all month, not doing it live streams, not making a big deal about anything. Parchment. Task Force, Darkened Hair. bloke could suggest one fine day. So I believe I'm going to save the CERN for tomorrow night because I want to really have some time to get into that. And we've already gone. The occult members are not sane because he lived. We see on Bible God. Earth in Invisible Cage. Earth in Invisible Cage. She's right around the corner. Sue Monsanto for limb loss. Spirit of Eternity. December 7th God. These politicians only lie. Lord God of Heaven, it's too late, far too late in the game. Consecrated chosen ones born to kill. For Frigga, consecrated chosen ones born to kill. Shake weight. Ghost is a melting pot of friendship. October 10th. Aries Cancer Crystal Bryan. Tiffany Christine Back to the Earthly Paradise. The 8th of June. Meme Warfare. Keen Perception of Reality. Donald R. Carson Jr. Vril Hidden Secrets or V Hidden Secrets. A. Justin Bieber. Bill and Melinda Gates arrested in Seattle. O. Long Johnson. Trump marks Istanbul. Eugenic is love. God who heals. James D. Dixon. Background actor narcissist. Sophia protect all the messages of God. Awakened One. Stony. Answer Me. Angelic Beast. Shadow Man. Chad Rogers. Dr. Stern. Black Suit. Cheesy Wolf. Diplomacy. Saxony. Who Are We? Guyver. Red Devils. Human Apes. Sine Wave. Like I've been working with frequencies. Sine waves. Day of Doom. Cancer Sun. Dutch Boy. I just keep going because it's interesting to me reading all this shit. It's related, you know. Yom Kippur 19. There's Decode the Secret Formula of Gematria again. Globalization is a falsehood. Embrace the truth. Embrace Christ. Holy hell, Megan Fox is old. False promises of the liar of the beginning. Robin Henry teases a demon. Insolence based on a lack of context. Triple H is horrible, horrible, horrible. Non-human entity named. Non-human entity named. Adults are funnier than kids. Murderous and arrogant. The Wooza and the Lat. I think you're Satan. When did I escape? King Eric John Christ. I'm right. Parallels have meaning. Parallels have meaning. Like the 143 parallel of this 1143 here. In the gematria, parallel shit. Infinity, the unlimited. Sensing other dimensions of the mind. Masonic electric darknet message board. Masonic electronic darknet message board. This person is John Cena's biggest fan. Suffer to the low cost. Low cost. God is whole. One. Boeing ceased state a Starliner orbital flight. In the war, lift up your head. Donald Trump target practice. Donald Trump is mind controlled. Excellent. Donald Trump is mind controlled. That's some stuff off of site. Tax them Labor Day. All. Soldiers and police officers unite. What's been happening? Isaac Price, Sword Beach. Some people call it Big Bang. Others call it God. Taurus, I love college football. Who is Io? Train ride. Elixir of. Improved Gate of Judah marketing. Corey Reed. Human garbage. The pagan god. Everglades. Candia family. Cronus. Nephilim. These are getting down to the 300 searches. Haha, we won. 8 days. 8 days keeps coming up. It's getting 21 to 21 searches. It's kind of getting off. Panheimer, heard of her. Northeast Neighbors, Salem, Oregon. I'm addicted to geometrics.org. 17 search results for that. Time wave. Bowsy Buff. The Death Bat. Anyone else feeling a time wave? Sirius C has been really, yeah, Sirius C in Orion. Right? Is it in Orion? Joshua. And I'm going to go to the last. No, I'm going to go to 13 here. Human gods break promises often. Human gods break promises often. The Matrix has to do this. I want to go home. Corruption in Atlanta, Georgia. Last lap of the Mandelbrot set. Northern Costa Rica is the Garden Eden. You need to come back to me. Chosen One's Crush Past. Demon Time. Cheech and Chong. Brian Adams. Hidden Sun. I Come in Peace, a Peaceful Man. Tuna Fish. Poonwalla. Poonawalla. More Proof Found, Princess Diana. God, my spouse to be vaccine. Oh, I can't say that one. Born September 14th. Freemasons are sacred of the ghost dance. Freemasons are scared of the ghost dance. You guys know what the ghost dance is? The natives up here did it, the ghost dance, to try and get their land back. Metaverse Facebook. Right there it is, guys. 7 searches for it already. Decode Heaven's God's a Man. A Divine Triangle. Restore Honor and Decency. Be a Secret. Rothschild borrows as strong as an octopus. Ladybug is Deutsch. I'm the living sacrifice. Zion is Elon Musk. To expose the deep state. I got Deep State Watchdogs. Inauguration Day. Love for all animals. That's me. Manticore. Feel better. Decline name ID. I'm not God. Turnip. Uncover. Israelite. The beloved. My dreams. In and out. Chimets. I am eternal. J. Trump. And stay dead. My life is no going back. Duke of Earl. Iron Attack. On the mend. I believe it. Goldie Hawn, I dare you. I bet the algorithm is just tripping out to what I'm fucking saying. What's the live stream doing? But the algorithm's just fucking just going insane. Oh, Project Veritas, they could be. Project Veritas could be controlled opposition. Barack Hussein Obama Antichrist. There's Barack Hussein Obama. Omega God on Earth, the Rogue Titan. The final Mickman. Enough is enough. Burn in hell. Better yourself. No Shadow 4. Masking children as child abuse. Do better. Wormhole 1. I cut my little finger. Ladybug tired of ciphers. I like Trump, man. Y'all fired. Great storytellers. Stressed out toilet pilots. Mom, I forgave Tyann. Submarine communication. Scable 666. Submarine communications Able 666. Frigga. Able 666. Mechanized satellite comet. Mechanized satellite comet. Circles, triangles, squares. Obamacare money laundering. Schizo is an English disease. Oh my God. Delete 1,000 prisoner. Kyle Russell. TikTok. Quentin cannot get an erection. Raymond God 66 Lost Mover. Androgynous Azari. Down in Africa. Hey, prepare yourself. Crash helmets at ready. I am Madison the Cipher. Users, but I am God of man. Make my day. Ancient man. Stupid. Accuser is. Biomagnetic. Robotoid. Frigga. Robotoid. I'm just gonna stop. Man. It's like, yep, I'm anymore, and I'm just what do you call it? Being blasphemous against the almighty gematria gods. Or the creator of the universe, the creator consciousness. This is what I mean. We're looking into the weirdo 19ES gematria and pulling. I'm correlating this stuff together. Maybe I've been up too long. I think it all goes together, but numerically and it's going along with me, some of my personal stuff and some personal stuff with other people I know and the country, and Crystal is faithful. Right there you go. There you go, Joe. Falcon flues for the 9th time tonight. Game over, Popes. Best lap dance I ever had. Scared Trumpy. A woman of God. Decode angels to heaven. He thinks he's me, DFU talking about it. Signet ring of Harry Melly. Capital Win. Forest Tent Caterpillar Moth. Decode Marcus Phillips. Operation. Remove false gods. No future for red hair elite. Crystal and her 3 kids. Cemetery Gematria 8. Mechanical inversion. Matrix glitches look like this. Fucking Leslie. The third search in This is Jewish, I believe. Jewish, third search. I am here to teach y'all all a lesson. Elephants can't talk, silly. Mechanical inversion. Septenary Gematria 8. Bearded Dragon, Evil Genes, Daily Beast, Roll Race, Theophile, Joe Morgan, Ping Pong, yours, bin number. Give me a hug. A new song. Oligarchy. Dear woman, they're telling us the deer have COVID now. Death is life. Then again, I know someone that says death is life all the time. As Azrael, law officer, Brady Bunch, Illuminato, hard and wet. It is real shoes deal and sold sides. Trying to pet a beast. Dakota, I really don't care, do you? A quantum artificial intelligence. A quantum artificial intelligence. That sounds something that, like, I just made up some quantum words today. Creator, no duality. I'm not mad, I'm hurt. There's a difference. Mind sweep. Mobius. Basic reproduction number. Plasma to hit Earth October 1st, 2021. Now, during October, we did have a lot of plasma hit us. All of this on Gematria looks good to me. 7 brides. He killed right Facebook off MMA. March 19th. Psychotic gangsters. No one can see me and live. Mushroom, my name is. Like a good neighbor, State Farm is there. Amen. Lover of all animals. Someone beautiful from Tahoe. There's no secret except. Oh. God's in the view, Carson Van. I'm so ready to be married. Decode Orlaw. He completely kill off flatter. Okay, now it's kind of getting. Ninth Gate, License of God, J. Epstein. Going to bed. How many of you are going to bed? Materials, because we're almost done here. Runic Magic, Mortals. Light Rain, it was raining earlier here. Aben Cooper. Whitehall. Jabba the Hutt. Dan Connor. Ode to John. Luciferian. CME Group. God Avatar is rich. Melchizedek Blue Rosa. Honey Caramel Eyes. Incarnation of Archangel Zadkiel. Balance is the divine right. Incarnation of Archangel Zadkiel. Mental parasitic terrorist. Nasara forever. Michael Joseph Hallett. Christ's son George, born Pisces. Neoliberalism fans, the flames of neo-fascism. Hello in days, soulmate. Thank God for Verb. Elizabeth is smoking hot. Satan itch much toy. Satan itch chum toy. Satan touch my hand. Oh my God. A bad day. I am the light of my soul. So there's some humor left in the world. Sea of salt. End of Islam. Before time. Night Veil. Drunk baby. Rage quit. Caveman. Murders. I named Jeff. Accelerating. I saw them. Gold fiber grooming. Theta fuel. It's pretty interesting shit, man. All right, we're done with the Jewish, and this is the last one I'm going to cover. God of Rome, Euronet, Self Healing, Enos Enoch, Frank Graham, Mean Girls, Federal Beast, Gold Mines, Heinz Field. I am Esther, sweating. Oldenburg, like Oldenburg, time of day. Powhatan, high as a kite. Thermite. I would have been high as a kite. My buddy would have showed up today. There it is, where it lives. Yeah, the feds kicked in his door. Give me a hug. All right, well, it's 2 hours, 42 minutes. We got time for the music I had set up. Well, it's just 3 minutes. So did y'all have a good time? Did you learn anything? Did you guys learn anything tonight? That the COVID is got a CRISPR in it. It's CRISPR. Go figure, huh? You know a Jeffrey George from your past, Frigga? Yeah, there was a lot of shit in there that was ringing your bell and my bell, for sure. Probably everybody in here got hit in that gematria. I imagine. Twas good, white boy. Twas good. I need a cigarette now. Again, I guess my buddy's not coming over. He missed out because I'm fixing to take all this shit, close it out. It's going into the tab collections in my tab collections. I'll be able to just hit a button and it pulls all these tabs right back up if I want to do a repeat show. You know, hopefully this video doesn't get taken down because I'm going to leave it up. I think it's important. I think everything I've shown here is really important. Really important. And remember this IBM RX for chemistry. This you use that. and you use this in correlation with it. Oops, I didn't mean to post that big of a link, but anyway, that's the result of one of the RxN mappers. And then you need the IBM RxN for chemistry, and then you need this link here, really. And then you need to get an account with I'll just go to search. Okay. There's PubChem site. Thunderstorm coming. Thunderstorm coming. We'll have to take a look at that. There's the PubChem, and then you need. Where's the NIH? You need PubMed, PubChem, and PubMed. Where's my PubMeds at? I had PubMeds up here somewhere, and Drug Bank, and oh wow, who really gives a fuck? You guys, I posted all the links earlier. They should be there. No reason to clutter up the chat again. I didn't do a good job. I kind of chopped it up, dude. But you know, if I recorded it, if I were to record it instead of doing it live, it'd be better. I do a lot better recordings, I think. But thanks, Soup. No, I mean, I try to make it friendly. Weird Hal Yankus. All right, here we go. Where'd my other YouTube video go? There it is. Damn ads, they've been doing this to me, man. Good night, guys. We'll do this tomorrow with the CERN tomorrow, okay? Or in the morning. Maybe in the morning.
Music.